Hi guys, welcome to another Dawn Academy video. Today we're going to be talking about neurons and the nervous system and everything that has to do with that. Okay, so let's start off with what is a neuron? Well, a neuron is a brain cell or a nervous system cell. Its job is to carry electrical impulses and everything that has to do with that. So how does it look? It's gonna be a little goofy drawing. Okay. So over here we have dendrites. Dendrites. So these are the ones that are going to be catching all the impulses, and so everything's going to start from here. So if we have our little impulse, it's going to be coming from here. And it's going to go through the dendrites across this, which is going to be our axon, across the axon, and it's going to continue up to here, which is going to be our synapse. So the dendrites are long and they're like kind of finger-like to be able to catch all different impulses. And okay, let's zoom into this a bit more. And then we get to the axon. The axon is special because it has a little covering on a few of the parts, and it's made out of a substance called myelin. So these are called myelin sheaths. Because they have lipids on them and because the electric impulse is polar, it can't really go through. So it's going to come here and it's going to be like, oh, what do I do now? So it's going to hop. And then it's going to hop again, then it's going to hop again, and it's going to hop again. And this is important because it characterizes the very fast speed of the electric impulses, because we really need the speeds very quickly. And if it has to go through here, it's going to take way longer to pass through all of these non-polar myelin sheaths. So it's going to hop on these little unmyelinated parts of the cell. And yeah, so once the impulse hops to all those different parts of the across the neuron, eventually they're going to get to the synapse. In the synapse, we have these little vesicles. So these are going to be our vesicles. They're made out of the same material as the cell membrane and what's in them is more important, which is the neurotransmitters. So we're going to have a bunch of neurotransmitters in all of these little vesicles, and this area is going to be called the synaptic sac. So this over all of here, this is going to be our synaptic sac. And so what's going to happen here is we're going to come back to this, but let's go to our action potential because we need to explain this in terms of where all the other things are coming from. You'll see what I mean. Okay, so in our action potential, we already spoke about how we're getting the impulse. We're getting the impulse right over here. So when we get the impulse, this is going to be called, let's mark our baseline first. It's going to be our minus 70 MV. This is going to be our baseline. So wait, let's go back to this drawing for a second. Before we receive an impulse, we're going to be just chilling at baseline, at here, at 70 millivolts. Though, when we get the impulse, do I have yellow? I do have yellow. When we get the impulse, we're going to shoot up. And this is going to be because, first of all, what's happening over here is that we have to accommodate the impulse. So inside the neuron, we actually have negative charges. Well, not negative. They're just, the inside of the neuron is less positive than the outside of the neuron. And that's because we have all our sodiums out here. And when the sodiums are out here, we're going to have a less positive charge inside the neuron. So how are we supposed to conduct the impulse if we don't have any charge? So what's going to happen is that the first step To helping us make this area inside more positive, we're going to have our little Na plus gated channel, and it's going to open. So what's going to happen when it opens? All the sodiums are going to come into the neuron, and that is going to give us our positive spike. So if you want to characterize this, we can say Na plus gated channels open. And it's also called a phase of depolarization. Depolarization. And that's because we're getting more positive. And this shows on the graph because our graph is going from -70 to a higher number. So it's getting more positive. So this is what we're seeing inside of the neuron. That's exactly what this graph shows us. Okay, so after we do that, we're going to get to some kind of peak. Let's do this in green. We're going to get to some kind of peak over here. And then it's going to start going back. We're going to start going back down. So what does that mean in terms of this neuron? So what's happening over here? Why is the charge inside of the neuron going back down? So after the Na plus all come in, it's going to become more positive inside, which is the phase of depolarization that we saw. So now we're going to have our Ks. So before this, our Ks were out here. Our Ks were in here. Oh, not Ka. Sorry, K plus. So now we're going to have our K channel. Oh my God, I keep writing Ka. We're going to have our potassium channel. And what's going to happen is that the potassiums are going to leave. They're going to leave the neuron because we want to make it negative again. We don't want it to be more positive. We want it to be less positive. So what's going on here in this dip in the graph is we're going to have K plus gated channels open. And because we're losing positive ions, the inside of this neuron is going to become more negative and less positive. Let's clear that out a little bit. So we're getting more negative again. Not really negative, but less positive. Which we also see because the graph is going down, which means we're lowering back again. And let me just write that this refers to inside of neuron. So first we have more sodium, so we're getting more positive. Then we have less potassium, so we're getting less positive. Okay, so now what's going to happen is that we are going to continue losing potassium, and a little bit too much actually. We're going to lose too much potassium. And why is that? Because if we go back to baseline too quick, then what if another impulse comes? Or what if the impulse is still here? Then it's going to become positive. It's going to become more positive again, and it can hop backwards. And we don't want that. So in order to prevent this, we go way, way below baseline because we don't want the neuron to have the ability to hop back again. So we go super, super negative, which is called a phase of hyperpolarization. So we're getting mega negative. Well, mega less positive, to be politically correct. Okay, and here the K plus channels are still open. Oh, and here it's called repolarization because we're getting back to the less positive polarization. Hyperpolarization, or it's called a refractory period. Refractory period. Okay, so after that, we're going back to our super, super, super negative. Super, super less positive. And then we're going to have another bud over here. And this is our well-known friend, K plus slash Na plus. Sodium potassium pump. So what's going on now? Well, as we said, the inside of the neuron is going to be super less positive because we're way, 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 way below baseline over here. So we have the wrong distribution of ions. Now all our K pluses are outside. Oh, when we had that phase, all of these left. Let's clear her up again. Okay, let's make it a bit more clean. Okay, so after our Na, we're gonna do a quick recap right now. So first of all, our Na, our sodium pumps opened, and the sodium flowed from outside to inside. So we have a bunch of, sodiums in here now. Remember, that was our phase of depolarization. And then, once we got to our peak on the graph, we were, let me show them both. Yeah, so first our Na came into the neuron and we had a more positive charge. And then we got to this peak over here and we started going down because the potassium gated channels opened. So our potassium went from inside to outside. So we have a bunch of potassium out here. And after that, we got to a phase of hyperpolarization down here. But look, the distribution of ions is not like we started. Although we could be going back to baseline, we don't have the right ions. The sodium belongs out here, and the potassium belongs in here. So what's going to happen is that this sodium-potassium pump is going to take the potassiums, bring them back inside against the gradient, and take the sodiums and bring them outside, also despite the gradient. What does that mean for our graph? Well, we're going back to baseline. And that is how electrical conduction happens in our neurons. I think we recapped that a lot of times. So if you need more clarification, just zoom back in the video. Okay, so that's what happens over here. Let's zoom back into our little synaptic sac. So after we had all of our conduction happening, our electrical impulse gets to the synaptic sac. Over here, we have another gated channel, but this time, oh, that is a very horrible box. But this time, it's for calcium. It's our calcium gated channel, and it's gonna open up. And the calcium is gonna go from out here to inside the sac. And once it gets super crowded in here, we have all our calciums in here. Again, they're going in through facilitated diffusion. They're going to start pushing these. So let's say this calcium is over here. It's going to go, hey, get out of my way. Hey, get out of my way. Hey, get out of my way. Until it gets to, oh my gosh, until it gets to the end. And once it gets to the end, what's going to happen is that the vesicle, remember we said it's made out of the same material as the cell membrane, is going to fuse with the cell membrane. And eventually, it's going to get these little neurotransmitters that are inside it, and they are going to come out of the synaptic sac. So we're kind of going to have like a little empty gap over here right now. We're going to get all the little neurotransmitters out of the sac, and they are going to be expelled into the synaptic gap over here. It's going to be our synaptic gap, synaptic cleft or gap. And once the neurotransmitters are in here, it's going to be our neurotransmitters. So all these vesicles are going to do the same. They're going to fuse with the cell membrane and expel their vesicles out here. After that happens, we're going to need to bring her down a little bit. Don't want her to, we'll bring the graph down actually. So after that happens, we're going to have some neurotransmitters just floating in this synaptic gap. And luckily, we are going to have a post, so this is going to be our pre-synapse. Then it's going to be our post-synapse over here. Pre-synapse or pre-neuron? Pre-neuron. Anyways, we're going to have a pre and a post. Might be neuron. Pre-post. So we're going to have our receptors over here. It's going to be our thing. We're going to have our receptors that are ready to catch all of our little neurotransmitters. So what the neurotransmitters are going to do is that they're going to diffuse across this little space. They're going to diffuse until they hit a receptor. Each neurotransmitter is going to get into a receptor, and that This action of the receptor binding is going to stimulate the post neuron to open its sodium channels, and look, the process repeats itself. Same thing. It's going to get an impulse, and then the impulse is going to flow through this neuron. It's going to get these to expel their neurotransmitters. Neurotransmitters are going to bind to the receptors, and the receptors will then stimulate the sodium gated channels to open. Sodium will go out, potassium will go in, and then the process will continue. And yeah, but we're not done yet, because sometimes, like now, we're gonna have some leftover neurotransmitters just chilling in this gap, and they have no receptors to bind to. So what's gonna happen is that if we're talking about acetylcholine, we're gonna have acetylcholine esterase, and it's an enzyme that's going to break down all of these little transmitters, and then they're going to be sent back into the synaptic sac, and they're going to be remade into neurotransmitters that can be used again. And yeah, that is basically the story of the neurons. So we got neurons, we got action potential, and we got neurotransmitters. I'm not going to go through the neonicotinoids, I think that's what they're called, but those are quite simple, so just take a look at the presentations, and that'll all be there for you. Thank you for watching. If you have any questions, let me know. Bye.